Hi, I'm Andy from the University of Washington, and today I'm going to be talking about strategies for properly controlling false discovery rates when a subset of peptides and a sample are relevant. In standard mass spectrometry-based proteomic data analysis, experimental spectra are searched against a database of peptides. These peptide spectrum matches, or PSMs, undergo target decoy competition to allow us to estimate FDR, or false discovery rates. The output of this process is a list of confidently detected PSMs. An implicit assumption in this process is that all peptides in the sample are relevant to downstream analysis, and by relevant we mean interesting. However, there are scenarios where researchers may be interested in detecting a subset of the peptides in the sample. For example, they may be interested in detecting peptides from a single protein or detecting peptides from a single organism found in a microbial community. The specific scenario that we are interested in is where the number of relevant peptides in the sample is a very small proportion of the total number of peptides in the sample. One use case we explored is the detection of the protein toxin ricin. Ricin is a protein toxin expressed in the seeds of the castor plant. This use case is interesting as it is illegal to possess the ricin protein, but it is legal to possess both the castor seeds and the castor plant. And there are many legitimate reasons to have these things. For example, castor seeds are used in the production of castor oil. So for the purposes of law enforcement, they're only interested in detecting peptides from the ricin protein, and they're uninterested in detecting any of the peptide from the castor plant proteome. To give you a sense of the scale that we're dealing with, ricin has about 80 relevant proteins, while the remaining castor plant proteome has about 2.3 million irrelevant peptides. So there's been several methods that have been proposed in the literature for calculating the false discovery rates on a relevant set of PSMs. Due to time, I won't be able to discuss the details about all of these four methods that we looked at, but we can give you some idea of what it is. For example, the second column shows the database that is searched, whether it is relevant and irrelevant or just irrelevant peptides. And this third column here shows you kind of the various steps that are shown. For example, in search and select, there's a database search, and then those PSMs that come out of the database search then go through FDR control. Out of the FDR control, we have a list of confidently detected PSMs, and then we filter out any PSMs that match irrelevant peptides to get us our confident list of relevant PSMs. So I won't be able to show you the data for this, but we find two methods, search and select and all sub, are generally invalid. And they're invalid in the sense that they do not properly control the FDR when there is a relevant subset of PSMs in your data. Subset search is itself invalid, and it's due to a specific phenomenon that hasn't been explored before. Namely, it's due to the interaction of only searching relevant peptides and the presence of a large number of neighbor peptides. Neighbor peptides are uninteresting peptides that are similar to relevant peptides in the sense that they have the same precursor mass as a relevant peptide, and to the same relevant peptide, they share a high proportion of B and Y ions in common. We have an example shown here on the right where the top peptide is a irrelevant yeast peptide and the bottom peptide is a relevant human peptide. As you can see by these dotted dashed lines between the two theoretical spectrum, these two peptides share a large number of B and Y ions in common. In fact, they share about 47% in common. In addition, these two peptides have approximately the same precursor mass, and they only differ by 12 parts per million. The problem of neighbor peptides in the context of subset search, where you only search for relevant peptides, is that spectra truly generated by a neighbor peptide is going to match really well to a relevant peptide. And if you have a lot of these neighbor spectra that incorrectly match to relevant peptides, you're going to have a large number. Your false discovery proportion is actually getting greater than than your estimated false discovery rate, which means your FDR estimate is invalid. So with subset search being invalid, we are now left with group FDR. So group FDR does properly control the FDR on a relevant subset of PSMs. It, however, doesn't explicitly consider neighbor peptides. It does implicitly. Another problem with group FDR is that it has a large multiple hypothesis testing burden. This burden is due to the fact that our database consists of both a relevant component and an irrelevant component, and the irrelevant database is much larger than the relevant database. As a result, spectra truly generated by a relevant peptide could, by chance, match well to an irrelevant peptide. The bigger the irrelevant database is in comparison to the relevant database, The bigger that is, the more chances that this could occur. As a result, you're going to lose statistical power. So as a result of all these drawbacks of previously described methods, we have created a new method, filtered subset neighbor search, FSNS, that 1, explicitly accounts for our neighbor peptides, and 2, doesn't have the low statistical power that group FDR has. And the way that this process works is that you take your experimental spectrum and you do a database search against all peptides you expect to see in your sample, which include neighbor peptides, irrelevant peptides, and relevant peptides. After that, you get a list of PSMs. With those lists of PSMs, you're going to remove any PSMs that match to relevant or neighbor peptides, and so therefore you only keep irrelevant peptides, which these irrelevant PSMs then go through FDR control. And so now you're going to get a list of confidently detected irrelevant PSMs in all rejected spectra. You're going to take these rejected spectra and then do a second database search against neighbor peptides and relevant peptides. After that scoring process, you're going to filter out any PSMs that match your neighbor peptides so that you only end up with relevant PSMs. With this list of relevant PSMs, then they actually go through FDR control so that in the end you get your confident list of relevant PSMs. So FSNS outperforms group FDR. What you see here on the left-hand side is on the y-axis the number of PSMs FSNS outperforms group FDR by, with the x-axis being a range of Q values, with the solid line being the median over 53 runs, and the dashed lines being the 5th and 95th percentile over the 53 runs. And as you see, over the entire FDR range that we plot from 0 to 10, FSNS outperforms group FDR. At a 1% FDR, that increased FSNS outperforms group FDR by 16.2%, or about 26.5 PSMs. Looking at a second data set where we consider UPS1 relevant and yeast irrelevant, we see the same general trend, just not to the same scale, where FSNS outperforms group FDR. Alright, the key takeaways is that the standard FDR control process assumes that all peptides in a sample are relevant. And if you only care about a subset of peptides in your sample, you need different methods. However, all previously described methods either yield an invalid FDR estimate or have low statistical power. So we propose a new method, FSNS, that outperforms previously published methods. I'd like to thank my collaborators in this, Bill Noble, Yuri Kike, and Deanna Plubel. And I'll take any questions. Thanks.